Hello students, welcome to Learner's Planet. Now again we are starting the biology class and the topic we are discussing is microorganisms. Microorganisms we have already studied, we have already discussed that these are such living organisms which are so small in size that they cannot be seen by the naked eyes. Generally, they measure in the units of micrometers, but the size varies. So, these microorganisms, they could not be seen by the naked eyes. And we had studied the solution also in the previous sessions, that if they could not be seen and they are existing, then they could be observed with the help of microscope. And further when studied, it is found that they can be our friends as well as they can be our foe. That means they can be our enemies. Further, we had studied that there are different kinds of microorganisms. So what are they? First, they are bacteria, they are fungi, protozoa, algae and virus. This is a broad classification of the group microorganisms. Now, in the previous sessions, we have already studied about the bacteria and the fungi. So now today, we are actually starting with the protozoa. The specialty of this protozoa is actually hidden in the name itself. The word protozoa is actually comprised of two parts. The very first part is proto and the next part here is zoa. So in this case, the word proto, it means primitive, and zoa is actually, it stands for the animal background. The one which is similar to the animal is generally highlighted with the suffix zo, zoo or zoa. So this protozoa is actually the primitive type of animal. Hence, it has been written here in the very first sentence that protozoa, they are the group of animal-like organisms. So, now we have to find out that why are we calling them animal-like? Why are they or how are they similar to the animals? This is what we have to understand in the beginning. So, when you say something is similar to animal, you have to think about the characteristics. So, at this point, we are studying microorganisms and we are comparing them with the animals. So, organization-wise, we cannot compare it much. But property-wise, we can definitely. And properties are coming from structures. So, in that case, we have to understand the structure at cellular level. Because these protozoa, they are the microorganisms which are just single cell bodies generally. So, animal-like means that first thing, we should compare the mode of nutrition. How they take their own nourishment. So in general terms, the animals are those organisms which are dependent on other sources for their nourishment. Secondly, they lack their cell wall. They do not have cell wall in surrounding their cells. That means the outer boundary in such type of cell is just a plasma membrane. So the cell wall is absent. Further, they are actually having developed type of nucleus and they are developed. So now, if we are studying protozoa and we are saying that they are animal-like, then what we mean by this point is that the protozoa is dependent on other sources for its nourishment. And such type of organisms which obtain their nourishment from other sources, they are known as heterotrophic organisms. So these heterotrophic, they are obtaining food from their surrounding. Animals obtain food from other animals or from the plants. Plants, they can synthesize their own food. But in the case of protozoa, what they are doing is something little bit different. These are actually saprophytic. What we mean by saprophytic? Saprophytic means that the organisms which are obtaining their food from the dead and decaying material in their surroundings. So here we have introduced two terms and that should be very clear here. What are those two terms here? We can write it here, we can spell it out here for you. They are heterotrophic. So what heterotrophic means? It means that is the organism is dependent on some other sources. They themselves cannot synthesize their own food. So those who are dependent on other sources for their own nourishment, they are known as heterotrophic organisms. And this is one of the most important characteristics shown by the animals. And hence, it is when we are saying protozoa is animal-like, then same property is shown by the protozoa also. Now, in the case of heterotrophic, the sources can vary. In case of protozoa, they are actually saprophytic. Now, what we mean by saprophytic is that they are dependent on dead and decayed material for their nourishment. So this is the special property shown by protozoa, which is actually similar to animal. Next property we are saying is they are single-celled organisms. Now when we are saying any organism, it is single cell, that means they are made up of only one cell. So such type of organisms which are made up of single cell, they are known as unicellular organisms. Unicellular, those organisms in which the single cell is able to perform all the life sustaining functions. So this is again a special characteristic of group Protozoa. They all are unicellular organisms, independent unicellular organisms. Further, they are eukaryotic organisms. Now what we mean by the word eukaryotic? Eukaryotic, we can divide this particular word again in two parts, that is eu and karyotic. The term eu means true, that means they are, and karyo means nucleus. So such organisms, they are actually having true type of nucleus. Now what we mean by the true type of nucleus? It means that the organism is actually having chromosomes which is surrounded by the nuclear membrane, and these chromosomes inside this nuclear membrane is actually present or embedded in the matrix, a jelly-like substance that is known as nucleoplasm. This means what? That the cytoplasm is separated from the nuclear material by nuclear membrane. So this is the special characteristic of eukaryotic organisms. Now a picture should be very clear in your mind that if we are saying that they are having true nucleus and the nuclear material is separated from the cytoplasm with the help of membrane, then what should be the appearance here? We can say, like, if this is a nucleus, then this particular membrane, the outer part shown here, this circular part, this outline is actually a membrane. And further, if I am saying the chromosome is present inside, so the chromosome we have studied, at the time of cell division, it is present in the form of cross-like structure. The chromosomes, they form cross structures. So let us assume this here. If this is one part, then cross can be made in this way. This is what we mean by the chromosome at the time when the cell is under cell division. If it is not in the dividing phase, then how this chromosome will appear? It is just a mass of entangled thread. If you just loosen the thread and you just lose it, you will find a bunch of thread is lying over there. That should be the appearance. That is actually the appearance of chromosome during the non-division phase, in the non-dividing cells. So here we can say this membrane is actually separating this particular chromosome from the surrounding cytoplasm in the cell. Now further, another point is that these eukaryotic organisms, they are not only having this particular well-developed nucleus, but also the other organelles are also well-developed. They are having membrane-bound organelles. And such features, they all are observed in the protozoa organism. Now we can see the examples here, like amoeba, Peramecium and euglena. And many more examples are there, like plasmodium, like trypanosoma, and many more. So now, if amoeba is quoted here, then you know what is the specialty of this particular organism? Amoeba. It is a free-living unicellular organism. Now, the special property of this organism is that they are having a very highly flexible membrane, cell membrane. The outer boundary, that is the membrane, is flexible, and thus it can change its own shape. And this property of changing its own shape is actually a boon for this particular organism, because it helps this organism for various purposes, like locomotion, like capturing the food material, like excreting out the waste material, and so on. So they are having this particular advantage. Peramecium, this is another example. It is having a slipper shaped structure. If we assume the shape, it appears like our own footstep, or you can say like the slipper. This Peramecium, it is actually having this locomotory property again, and in this case, the locomotion, it takes place with the help of minute hair-like structures present over its outer wall or the outer boundary, and that is known as cilia. Further, euglena. Euglena is again another example of protozoa, and this euglena is actually having flagella for its own movement. Now, in the beginning we have said that protozoa group is actually heterotrophic and it shows saprophytic mode of nutrition. But in the case of euglena, the mode is little bit interesting. Normally they are heterotrophic, but if in their surrounding the food material is not available, then in that case they are having the chlorophyll. They are having the photosynthetic pigment, and thus they are able to synthesize their own food. Thus we can say they can show the mixotrophic type of nutrition. Besides that, we have quoted some other examples like Plasmodium, like Trypanosoma. So these Plasmodium and Trypanosoma are actually they are parasitic in nature. That means they are not independently living, they are not free living organisms. Rather, they are resided inside the host body. For example, Plasmodium. Plasmodium is actually an organism which is causing malaria in human beings, and it is found in the saliva of the mosquito. Which mosquito? It is female Anopheles. We can understand these particular examples quite in another interesting way with the help of these diagrams. So here this picture is of Peramecium, and we said this Peramecium is actually having a slipper structure. Its shape is somewhat like our slippers. So now in this case, let us start studying each part individually and just try to understand the features of the group Protozoa. Most of the organisms shows the general properties as we have discussed earlier. For example, which properties? They are animal-like, that means they are heterotrophic in nature. Then next, they are actually unicellular. Besides, they are eukaryotic, and they can show the locomotion. That means they can move from one place to another. For that purpose, they are provided with the special structures. So that's what we are now going to study in these particular pictures. In Peramecium, they are having this outer wall, this membrane is here, and this membrane is actually provided with the hair-like structures. And these hair-like structures are known as cilia. So the cilia, they are very small, very minute, and they are present in many number. The number is not few, it is present all over the surface of this particular organism. Now, the special property of cilia is that they can move to and fro. They can show the motion in upward and downward direction, backward and the forward direction. So this backward and forward movement direction actually helps in pushing the organism forward and backward. So this is how the cilia provides the advantage for movement of this particular organism. The next part shown here is mouth pore. Now this mouth pore is actually a kind of opening, and this opening actually opens behind in a cavity, and this small cavity is known as a buccal cavity. Mouth pore, you can imagine, mouth generally serves for which particular function? It is for ingestion. Fine, it is for speaking also, but actually we are studying this particular characteristic here. So these organisms, they cannot speak. So here the mouth is simply meant for the ingestion purposes. What we mean by the ingestion? It means taking inside the food material. So these pore actually serves for this particular function. Now these are little organisms. Imagine how they can take up and pick up the food and ingest, or it will put inside the buccal cavity. They are not provided with certain new hands or something. Rather, this particular purpose is actually solved by the cilia. The cilia, when it moves in up and down direction in to and fro motion, then it brings the surrounding water close to this particular mouth opening or the pore, and thus it will push the water into the buccal cavity. Further inside the buccal cavity, it is again provided with the cilia. So these cilia inside the buccal cavity, they will push the food material forward towards inside the cytoplasm, and by the diffusion and different ways, this food material will go inside the cytoplasm. How? In the form of a vacuole. At the end of this buccal cavity, it will receive the food material and it will enclose it in the form of this food vacuole. Food vacuole, it is for the having the digestive enzymes. Now these digestive enzymes, they actually help in the digestion of the food material. Food is present inside the food vacuole, and this is having the enzymes. These enzymes work over the food. They break down them in the smaller particles, and then these smaller particles or the digested food is actually utilized by the cytoplasm or the organism. Further, it circulates in the entire part here, and this movement actually exposes it to the different region. And meanwhile, it is releasing out the digested food material via diffusion through this membrane or the cell wall of the food vacuole. And then it is utilized, the digested food is utilized by different other parts of this organism, the organelles and other parts we can see. And then it is assimilated, that means it is converted into other useful material. And further, the remaining food which is left out here, the undigested food material, will reach towards the anal pore. You can see here this particular structure. This anal pore will actually remove out the undigested food. We can highlight here this anal pore. It is this structure, and when undigested food material reaches this anal pore, it will be thrown out in the surrounding water, and thus the food material or the digestion is actually maintained in the this unicellular organism. Further, the another part here is contractile vacuoles. These contractile vacuoles may be more than one in number, and they are actually helping in the circulation of the things inside the cytoplasm. Besides, they also help in the contraction and relaxation of this organism and helps in the movement also. So this contractile vacuole, we are saying it shows contraction and relaxation, so that is actually maintained by the water regulation system. So this contractile vacuole is actually maintained, or it is driven by the water vascular force. Further, there is another part here, it is macronucleus and micronucleus. So nucleus, we know it is the brain of the cell. They are actually enclosing the genetic information inside. And this is the special characteristic of Peramecium, that there are two nuclear parts in a single organism. One is larger, which we call as macronucleus. The another one is a smaller one, which we call as a micronucleus. And these are actually the control units. They help in the control of different type of cellular activities here. So this is about the Peramecium. We can more and more understand this protozoa with the help of another example, that is amoeba. So amoeba, we have chosen this amoeba to explain in this lecture. The reason is because they have the special characteristic. They are different from other organisms of the same group in terms of its flexible membrane. Here is the plasma membrane. Let us understand the features here. We can highlight it much more better. The arrow is for this. So this particular plasma membrane is the outer boundary of amoeba, and it is actually highly flexible, and this flexibility provides more and more advantage to this living organism. What advantage they can get? Because of this irregular shape, the locomotion becomes somewhat easy. They can move by changing the shape. Further, this property of changing the shape provides it advantage capturing the food material in their surrounding. Not only this, they can also excrete out by changing the position and removing out the food material from here. Now the next part shown here is nucleus. The nucleus is the brain of the cell, as we have studied just now. Besides that, we have highlighted here pseudopodia. Pseudo means false, podia means feet. So they are actually having, these are the false feet for amoeba. Why we are calling false feet? Feet because they help in the movement, locomotion. And false because they are not standard. They are not present permanently. So this is why we call these type of structures as pseudopodia. Pseudopodia not only helps in the movement, but also it helps in the ingestion process. That means engulfing the food material from its surrounding. Once it is engulfed, it will be enclosed in the vacuole, where the digestion is going to take place. Now we can show how the capturing is actually taking place. If we take the help of diagram, we can actually easily say that if these are the pseudopodia, then how they are actually working here, and this is the food material in surrounding, then it will get enlarged and it will just surround the food particle. So the diagram is going to be like this. It enlarges and it will capture the food. The food will come here, and then it will definitely go in the center region, in the cavity. Further, the end points are going to enclosed. So we can see the diagram here. It will appear something like this. These are the flexible parts of the, this is possible because of the flexibility of the membrane. When it touches here, then definitely it gets dissolved at the end point and the membrane remains like this, and the food particle will be captured inside. Now this is what we call as the food vacuole. And this food vacuole, it will be having the hydrolytic enzymes. These enzymes, they will help in the digestion of this engulfed food material. It will break down the particles, food material into smaller and smaller parts, and then those smaller particles are able to diffuse out of the membrane. Once it is diffused out, it will be utilized by the surrounding parts of the structure of the amoeba. There are certain parts which remains undigested also. So that undigested material, it has to be eliminated out of the body. So further, these particular material, it will be removed out, it will remain empty, and it will go towards the pseudopodia, it will get fused with that, and if the remaining part or the undigested food, it will be thrown into the surrounding. So this is how this amoeba survives in their surrounding. Besides this, there is a contractile vacuole. Now this contractile vacuole Again, it helps in the circulation of things inside the cytoplasm. Also, it helps in the movement site-wise. So here we can understand the characteristics of protozoa. What important characteristics are there? That they are heterotrophic, they are unicellular, they are eukaryotic, and they shows locomotion. And this locomotion is generally taking place with the help of various structures like cilia, flagella, or this pseudopodia. So now this is all about protozoa, and we are going to study about the next group of microorganisms, that is algae and virus, in the next session. Till then, goodbye and thank you so much. Just go through your studies. Have a nice day. Bye.